Hi, I'm Ryan, and in this video I will be discussing the genetic technology of gene therapy. What if I told you there is a cure for blindness? The Shea Eye Institute at the University of Pennsylvania has made a discovery that is showing promising signs. The patients they have been working with have a blindness called LCA, which is short for Leber congenital amaurosis. This type of blindness is caused by mutations in the GUCY2D gene. With this type of blindness, the photoreceptor cells are relatively preserved, which means that gene therapy could work to restore sight. What is gene therapy? Gene therapy is when you add a normal copy gene, also known as a therapeutic gene, to the genome. The hope by entering the normal copy gene into the cells is that it will be expressed instead of the non-functional mutant gene. How do you choose the right therapeutic gene? The type of therapeutic gene you use is dependent on the effects of the disease. For example, a disease that causes a gain of function needs a therapeutic gene that would combat the mutated gene and suppress it or inactivate it. LCA doesn't work that way though. The mutation that causes LCA is caused by a loss of function of the photoreceptor cells. The therapeutic gene would have to get the photoreceptor cells functioning again, so you would just make the therapeutic gene a wild-type gene. Methods of therapeutic gene delivery. There are two main ways to deliver therapeutic genes into the cells. The first way is called in vivo gene therapy. This process is easy to perform because it can be as simple as injecting the therapeutic gene into the eye or inhaling the gene into the lungs. The downside to this method is that it may just introduce the DNA into a small amount of the affected cells instead of all of the affected cells. The other way is called ex vivo gene therapy. In ex vivo gene therapy, the cells are removed from the body and are exposed to a large amount of the therapy gene in a culture and is left in it to grow. The cells are then returned to the body. In the case of LCA blindness, however, the type of gene therapy that was used was called AAV therapy. According to AskBio.com, AAV stands for adeno-associated virus. AAV is a virus that does not result in a human disease, and it cannot replicate itself without help from outside sources, so that means that scientists have more control over how much of the virus is given to the patient. One downside to this, however, is that AAV DNA slowly degrades, and the gene therapy needs to be repeated periodically to maintain a supply of the protein that is produced from it. What was the results for the patients with LCA blindness after gene therapy? For each patient, only one eye received treatment so that they could more easily compare results. Within 8 days, patients were showing vision improvement. The patients found a major improvement to their vision and were even able to make out objects in the dark. You may be thinking, gene therapy sounds great, but there are some moral questions that arise from it. One of the moral questions that comes along with gene therapy is if it should be allowed to enhance human traits like height, athleticism, or other traits. For example, would it be fair for someone who is naturally 5'8" to get gene therapy and become 6 foot? Another question is, because it is a high-cost procedure, is it fair that only rich people may be the only people able to receive the procedure? Finally, if gene therapy does become widely used, would it make society less accepting of people who have disorders or disabilities? People may look down upon disabled people who have not received gene therapy and think that there is something wrong with them for not receiving gene therapy, even though gene therapy may not help their disability. To quickly review, gene therapy is when you add a therapeutic gene into a mutant genome to make the mutant gene inactive or active depending on the mutation to restore normal function to the cell. Choosing a therapy gene depends on if the mutant causes gain of function or loss of function. There are two main ways to deliver a therapeutic gene, which is in vivo and ex vivo. With in vivo, the therapy gene is injected directly into the mutated cells. With ex vivo, the mutated cells are removed from the patient and grown in the therapy gene and is then transferred back into the patient. And finally, AAV therapy uses a virus to deliver the therapy gene and is easy for scientists to control because it is unable to replicate itself naturally. This article on gene therapy being used to treat people with LCA is proof that gene therapy is being used to help people with multiple disorders, and there is still so many undiscovered possibilities for what gene therapy can do. There has been research into gene therapy for cancer and is used to treat cystic fibrosis. Cystic fibrosis causes damage to parts of the body like the lungs and digestive system because it affects the way cells produce mucus, sweat, and digestive fluids. Normally, these fluids are supposed to be thin and slippery, but cystic fibrosis causes them to be thick and sticky and can plug up the tubes and passageways in the lungs and in the digestive tract. Obviously, that is not good, so now gene therapy can be used to help symptoms. Even though gene therapy is currently expensive, I am sure we will develop a more cost-effective process, or that insurances may cover gene therapy to make it affordable for people who need it, like people who have LCA. According to US News, LCA affects 1 in 40,000 newborns, which makes LCA one of the most common forms of congenital blindness. This shows us that some common disorders and disabilities can be helped by gene therapy and that it should be an option that is accessible to people. Thank you very much.